IICT
IIT
campus.
Mumbai.
Yeah.
Christian has also joined. Muhammad Tharjade has
Great.
also joined. Harish from Japan has joined. Lei Jian from China has joined. I see quite a few people from different countries who are IBA member. They have joined. We have
total 44
Share,
are already
Share
there.
Is also there. She's from Iran. And then we have somebody from Indonesia. Poonam from UK has joined. So people will keep.
So it's truly global.
Tejendra has joined from Indonesia.
So, Professor Ganguly, also beside this talk, I will request you to actually be part of the society, help us more and more actively. I am affiliated with this society since beginning. Dr. Pandey gave me this opportunity. So, okay. So I think we can start if everyone.
Yes.
So
I think Vinod is also there. Vinod, are you there?
Yes, sir, I'm here. Yeah.
Okay.
So I request, to the audience, very good afternoon and good morning and evening at different zones. It's pleasure for me to announce this is the 15th Global BRSI talk on the application of nanotechnology in biology and medicine. Actually, we started this global lecture series with the initiative of Professor Pandey. And Professor Dr. Ganguly is the 15th lecture, giving the 15th lecture. So Professor Ganguly is need no introduction actually. He is the director of ICER Behrampur currently, and he is also holding Professor K.N. Jha Chair in the Department of Chemistry, IIT Delhi. He was earlier Institute Chair Professor of Chemistry Department, IIT Delhi. He is also joint professor in the Department of Material Science and Engineering and there in IIT, and he was Deputy Director Strategy and Planning at IIT Delhi. He is known, and everyone probably know him by that introduction. He is founding director of Institute of Nanoscience and Technology in Mohali. Professor Ganguly obtained his PhD degree from Solid State and Structural Chemistry Unit from IC Bangalore in 1990 in the area of thallium-based high-temperature superconductor, for which he got Sudro Medal of Indian Institute of Science. He worked with Professor C.N. Rao for his graduate degree. He has been a visiting scientist at DuPont, USA, and a postdoctoral associate and visiting scientist at MS Lab, Iowa State University. He's also visiting professor at EPFL Louisiana in 2016. His area of interest are in the design of nanostructured material for application in water purification, solar energy conservation, and microfluidic devices and high temperature superconductivity. He has published over 300 publication papers and have 5 patents to his credit. He has successfully completed many important national research projects. He is recipient of National Award of Nanoscience and Technology given by DST Government of India, the Chemical Research Society Silver Medal, the Material Research Society India MRSI Medal, C.N. Rao CRSI Medal Award, Chemical Excellence Award by Indian Society of Chemists and Biologists, the Bangalore India Nano Award given by Karnataka government. He is also Fellow of Royal Society of Chemistry, London, Fellow of Indian Academy of Sciences, INSA Fellow, and NASI Fellow. And he also is Asia Pacific Association Fellow of Material Sciences. He is currently Associate Editor of Bulletin of Material Science and a member of editorial board of ACS Material Letters, Chemical Physical Letters, J Industrial Chemistry Society. So I think many more things to his credit. I would not come in between his lecture and you. So I think I'll request now Professor Ganguly to deliver this lecture on application of nanotechnology in biology and medicine. But before that, I'll request Dr. Pandey to, as a president, to say a few words, chairman of the society, to say a few words.
Thank you, Sunilji. On behalf of the society, I welcome Professor Ganguly for this lecture. As we mentioned by Professor Sunil Khare, that this lecture series was started by the Biotech Research Society India. However, now it is a joint global lecture series organized by the Biotech Research Society in association with the International Bioprocessing Association, IBA, which is having its headquarters from in France, and Professor Christian Larousse is administrator. And we have several members joining from this IBA. So this will continue now as a joint global lecture series being organized by one by BRSI IBA, other one by IBA BRSI, like that. So the purpose is to get acquainted the researcher all around the world, in particular the members of the societies, our societies, with one of the most advanced topics. And from that point of view, today's lecture being delivered by Professor Ganguly on nanotechnology in biology and medicine acquires great significance for all of us working in varied areas of biotechnology or biological sciences or material sciences and engineering. So thank you very much, Professor Ganguly, for accepting our invitation. And over to you. Forum is now for you, Professor Ganguly.
Okay, so I can start?
Yes, please.
Okay. Thank you, Professor Khare and Professor Pandey, and all the members of the Biotech Research Society of India and the global scientists who are associated with this association, IBA, for inviting me to give this talk. I am actually trained as a chemist and more close to materials. But during my term and tenure as a director of INST Mohali, I came to learn biology, interact with biologists more and more, and my appreciation of biology has really grown. And the area where a lot of things need to be done is, I think, more in biology and, of course, in medicine and diagnostics and all these things. And truly, I learned the interdisciplinary nature of biology with physics and mechanical engineering, etc. It is brought together by the subject of nanotechnology. And before starting, I just want to say that I'll give some basic introduction to nanotechnology, for there may be some students, I don't know whether they are there. And then I will talk about some problems of two of my PhD students, Manisha and Manoj Kumar. And this is also a global interaction. Manoj Kumar did a joint PhD with me and with the University of Queensland, Michael Wu, who is kind of biologist and materials and biologist. And Manisha is a joint PhD student with me and Professor Paras Prasad at University of Buffalo. So this is the kind of research I will tell towards the end is really international research with two groups, one in Australia, one in United States. Most of the work I did was at IIT Delhi, which I'm going to describe. And IISER Berhampur, I just joined 1 year back as a director. So my slides, okay. So all of us know what are materials. They are all around us. And we can see materials as small as 0.03 millimeters with our naked eye. If we want to see something smaller, then we use microscopes like the scanning electron microscope, which can see easily 150. Even today, the latest scanning microscopes can see 20 nanometers or so. So even smaller objects, say at the level of atoms, which is like 0.1 nanometer in diameter, the smallest atom, hydrogen atom, is around 0.1 nanometer, little bit bigger than that. Even those can be seen today by the latest microscopes. So the world of small things is really gone up because of the technological improvements in many aspects, especially microscopy. But apart from microscopy, there are other areas by which we can visualize, especially things like ultra-fast spectroscopy, etc., the dynamics of systems which are very small. So this is to appreciate what we are looking at, nanotechnology. We have to look at the length scales. So nanotechnology, typically we say things which are within 1 to 100 nanometers. Of course, it changes from subject to subject. In physics, nanotechnology is always in very small, say 1 to 5 nanometers. That is where physics, electronics, etc. talk of nanotechnology. Whereas in biology, even 200 nanometer, 300 nanometer particles, liposomes and all that, can be considered into in bio-nanotechnology. So as per the context, the size of nanoparticles or nanotechnology changes, and in physics, it is smallest. In chemistry, it is somewhat in between, say, 10 to 30, 40 nanometers. And in biology, the particles that we are normally talking are more than 50, 60, 100 nanometers. So I was telling you that today we can see even the atom, and actually inside the atom also with the latest microscopies like the one I am showing. This is a aberration corrected electron microscope, which in Indian currency can cost 35 to 50 crores depending on the accessories that you take along with this. This is was a revolutionary technology which was brought about by physicists who kind of showed how you can go beyond what in physics was a block, which is called the diffraction limit. That means you can see objects of size which is related to the wavelength of light. So if a light of wavelength lambda is used, you can see objects which are lambda by 2. But this diffraction limit, the limit of seeing small objects was kind of bypassed by those scientists, and this aberration corrected microscope was developed around 2005-06. It was developed and Nobel Prize was given, and today latest microscopes are available. So I will not dwell too much, but all of you know what is the relation between micron, nanometer, and etc. In why nanometer is important, many people ask why not picometer or femtometer, which are smaller than nanometer. But nanotechnology is more important. People normally do not talk of femtotechnology or picotechnology, although they are smaller than nanotechnology. One key reason is that laws of physics actually change at the nano dimension, and you go from a classical physics to quantum physics. So that is one reason why the nano size is very important. There are many other reasons what happens to magnetic properties, optical properties, etc., as you go to nano dimensions. So see, these are some of the simple examples, one or two I will say, which is known now for over 20 years, that if you take a small particle of gold, it can oxidize carbon monoxide to carbon dioxide. But if you take a normal particle of gold, it does not do that. That means catalytic properties are changing. If you see a butterfly which has two colors, this explanation of these two colors of the butterfly, which is called biophotonics today, or nano biophotonics, can be explained by the small pores within the wings of the butterfly. When the butterfly flies, the pore size changes and the color changes from brown to blue. There is no dye in the butterfly, like a blue pigment or something, but the butterfly's color is due to scattering of light from different size of the holes in the butterfly's wing. This is a nanotechnology in biology, in nature, which the butterfly has been given by nature to protect itself. So there are lots of such properties. One of the first examples of applications of nanotechnology was in cosmetics and creams. And L'Oreal brought out many of these objects in the year 1999-2000. One of the first objects was putting on a sunscreen lotion. The sunscreen lotion is basically made either of zinc oxide or titanium oxide nanoparticles. What you see here is the size of the nanoparticle. If you see 195 meter nanoparticle, TiO2 particle, if you rub on your surface of the hand, it gives you a white color. If you reduce the size of these particles to 60, 35, 15, you see the color is going away. But the property remains. What does the nanoparticle do on your surface? It protects you from the UV radiation in the solar light. So you wear it when you are playing or you are on the beach, etc. So depending on the size of the nanoparticle, the titanium oxide color is going from white to transparent color. And normally we would not like to look like a white face if it is possible for us to have this nanoparticle to be invisible. So this is possible to do by just changing the size of this nanoparticle. So these are some of the first applications of nanoparticle in cosmetics and in sports, where a lot of tennis rackets, tennis balls, etc. use nanoparticles for more efficient properties. In nature, we have nanotechnology. You know, these are magnetotactic bacteria. These are found in the oceans, where if you have small, when you look at these bacteria, you find these Fe3O4 iron oxide or magnetite particles in their body, inside the cells. And still the explanation is not clear how the Fe3O4 particle forms. But the reason scientists ascribe that nature has made these nanoparticles in this bacteria is that these bacteria are guided by the Earth's magnetic field because of these magnetic nanoparticles inside. And they can find the source of their food as a change of the ocean currents and all. And probably this is somehow this has developed naturally for these organisms to have magnetite nanoparticles inside. But I believe the exact reason is still not clear. There are objects which walk on water. That is also because if you take a cross-section of the legs, you will find nanofibers under their legs, and they can, air can be trapped within these nanofibers, and that allows them to give super hydrophobic properties. They can walk on water, etc. This is natural in scene. Lizards walk on the walls, ants can walk. All that is because of nano-structured properties in their legs. We have hierarchical nanostructures, and this aspect which we learned from nature, today chemical engineers use a lot in making hierarchical structures using polymers in the laboratory. So these are the polymers which have been made in the laboratory, which will give you similar properties which you see in the legs of the lizards, etc. This is now very common. This is already known for now 10, 12 years, etc. The super hydrophobicity of lotus leaves, again, is due to hierarchical structures. And this has been used in glasses to make smart glasses, where you can have these hierarchical structures seen in lotus leaf, and which does not allow dust particles to settle on the glass. And you can make smart glasses because of these hierarchical nanostructures. You can make them on shirts, so they can be made quick drying, etc. And many such shirts are now available. It's even made in India by a company in Bangalore. You can make different structures which can be used for the ships and submarines, where you have nanostructured polymers which mimic the surface of the skin of a shark, because it was studied that sharks never have bacterial infection, or molds or fungus do not normally take place on the shark surface. And scientists studying the surface of the shark found out that there are structures on the surface which, if you can mimic using some polymers, then you can see, as the left panel shows, this is a polymer with nanostructures. The right panel has the same material with no nanostructures. But if you apply some bacterial colony, few cells, they grow into several large colonies of bacteria, whereas on the left side, there are much less bacteria. So this is due to nanostructures. You can prevent bacterial colonization. So coming to more today's talk, that what can technology, where it can be applied in biology, medicine, you can apply in drug delivery, targeted therapy, imaging, molecular imaging, disease diagnosis, gene delivery, molecular marker identification. In all these areas, you can use nanoparticles and nanotechnology in different ways. On the right side is given some aspects of people using anti-cancer vaccination, antibacterial vaccination, which is possible today. I'll talk a little bit about it. So one of the key things that in biology, if you see the size of objects which are of use in biology or medicine, you can look at the size of antibodies, which are around 10 nanometers. Viruses are around 100 nanometers. Bacteria around 1000 nanometers, which is 1 micron. And then cancer cells are around 10 microns or 10,000 nanometers. All right. So this size is important to keep in mind when people are designing some kind of technology with various cells, with various liposomes or drug-carrying dendrimers or polymers, etc. So a typical drug delivery through a polymer nanoparticle can be like this, where you have a polymeric nanoparticle where the drug is encapsulated, and you have a receptor molecules on the top which can bind to certain receptors of the particular cells which you are targeting. So typically, you have liposomes, which you see you have molecules which are like surfactants. In chemistry, we study surfactants which are having a head group and a tail. If two surfactant molecules are connected in a manner, the way it is shown here, like a bilayer, the bilayer has the head groups on the outside and the tails inwards. So these are called liposomes. So these liposomes are there in our body also, and many membranes are there in our body, which are similar to this kind of two surfactant layer or bilayers, which are available. They may be flat, they may be circular. There can be vesicles also on similar things. And you can have drugs or other molecules entrapped within this. So this is one of the primary mode of drug delivery or gene delivery that many scientists have been working over the last 15 years or so, 20 years. Little bit more detail on the history of the development of such drug delivery units. So the first liposome was discovered in 1964. So you see 1964 to nearly 30 years, people have been developing, a lot of organic chemists have developed different polymers, and first develop the polymers and then try to control their shape, control their stimuli response. That means certain polymers, if you can allow them to open up under certain stress, it may be pH, it may be temperature, it may be magnetic field, it may be UV radiation. So a lot of research in these 30 years people have done in trying to have a controlled release system when the polymeric system will open up and release the cargo inside. And the first controlled release polymer, as you see, was designed in 76. The first published dendrimer in 78. First targeted liposomes in 80. And of course, there is a very important effect all of you will know better than me, probably as biologists, which is called the enhanced permeation and retention effect, the EPR effect. So that means you want the drug to be in the circulation system for long to reach the targeted cell. So if you can have these nanoparticles with an enhanced permeability and retention, then you can have the drug much more active at the right site where you want to. So this effect was discovered in 1986. And then people designed long circulating nanoparticles. There was the first drug, Doxil, approved by FDA in 95. There were other drugs then subsequently which were approved. Today, probably around 12 drugs, cancer-related mostly, are approved by the FDA and are there in the market. So there are these drugs you may be heard of: Abraxane, paclitaxel, Doxil, doxorubicin. These are common drugs which people have worked for over last maybe 15, 20 years, and how to encapsulate them in different polymeric nanoparticles, different liposomes, and then have some markers or molecules on top which can target certain receptors, etc. So nearly 50 years of research has been going on in this area. Other than polymeric drug delivery units, like what I discussed, liposomes, polymerosomes, you can have dendrimers. You can also have a polymeric micelles. You can have inorganic nanoparticles like silica nanoparticles or iron oxide nanoparticles, gold nanoparticles. There is not a very large library of inorganic nanoparticles because they may be toxic. What has been found more or less, silica, iron oxide, and to some extent gold nanoparticles of certain size are the best which can be used in the biological systems without toxic effects. So a lot of research in inorganic nanoparticles has gone around silica nanoparticles and different types of silica, and I will give you some of our work on silica nanoparticles and the size and shape which are effective in drug delivery, etc. But there is a lot of other people's work in iron oxide based nanoparticle, especially using magnetic fields and increasing the local temperature by using these magnetic nanoparticles to kill cancer cells. There has been a lot of research on gold nanoparticles because gold nanoparticles of certain size and shape can be excited using near IR lasers, and they can raise the local temperature again by 4 or 5 degrees, and that is sufficient to kill the cancer cells. So there are gold nanorods which are very effective, and one of the first drugs which was FDA approved was made, I'll show, by Naomi Halas at Rice University, Texas, where she first designed this kind of gold nanoparticles. There are these different polymeric, inorganic, and lipid-based nanoparticles, which are primary interest in biology and medicine, which most people work on for drug delivery, etc. So drugs with very low solubility will have delivery issues. So how to make them more soluble so that they have increased bioaccessibility, and how to make more quantity for intravenous intake, etc. So these limitations can be overcome by nanotechnology approaches, and some of these we will discuss, especially by using inorganic and polymeric nanostructures, including dendrimers, or even using silica nanoparticles of certain shapes. So mesoporous silica nanoparticle-based carriers have been of lot of interest. It increases the loading capacity and the controlled release of the drugs by using some modification of the surface hydroxyl groups on the silica. If you can modify them, then you can have a controlled release of the drugs, etc. These are 4 examples just from literature. The first example is the delivery is through the nasal route, and you have nanoparticle modified surface with different peptides, and they can be taken from the nasal route and across the blood circulation. So these can be directly given to the brain to treat diseases with like epilepsy, Alzheimer's, etc. The second one is where you are using a nanoparticle with drug and to enter into the stem cells, the mesenchymal cells. And this is more for bone tissue engineering, etc. You can also use nanomedicine, etc., for using the directly into liver cells. And also you can, the last one, the fourth example is what a lot of research has been done on targeting tumors, etc. So this is some idea of what size and shape does in when it enters the bloodstream. So if you see that a particular size and shape, if you have, say, rod shape, then it moves out of the vascular structure. And if you have a certain size, then it on the right top, you will see that it has got more clearance. So the size is small, it has more clearance, especially if it is charged. The other thing is that if you have coated nanoparticles, it behaves in some manner. And of course, if you have targeted, that means you have certain moieties on the surface which have specific molecules, then you can target better. So there are different types of particles you can design: spherical particles, rod-shaped particles. You can even design triangular particles. You can design core shell particles, that is coated particles. And you can design particles which have molecules on the surface to target some receptor molecules. As I said that you can release the cargo from particles under certain stimuli. It can be light, it can be pH, it can be enzyme, it can be magnetic field, it can be temperature. So on the right side, you can see there is a particular molecule which is zinc oxide in organic system, where you have attached doxorubicin, and then we have created a metal organic framework using 2-methyl-imidazole. So this is like what inorganic or material chemists study. It has been found that rather than use zinc oxide, zinc ions are good because they create reactive oxygen species. Reactive oxygen species can rupture the cell membranes. So zinc ions are very useful in rupturing cell membranes. So when you attach a zinc oxide with the drug, and if this zinc oxide can be protected, then it can be effective to reach the target. So that protection here is being shown with the what we call is a metal organic framework. Here the metal organic framework is ZIF8. I'll talk about ZIF8 later when I explain one of our recent works on a metal organic framework based drug delivery system. So this is about stimuli responsive molecules you can generate. One of the examples from modern, which has been very effective in developing a vaccine, is the mRNA-based Moderna vaccine, where it uses a liposome. So this is an example of a liposome. So where you have your messenger RNA inside the liposome, and this liposome actually, if you see the right panel, on the left side is a standard liposome. On the right side, you have got a liposome which has got some molecules on the surface. So you design molecules. So you have regulated it. You can attach an antibody, you can attach a protein, you can attach a peptide. So a lot of these things you can attach on the surface of this liposome. And based on this, and your the DNA or RNA, the genetic material is inside and encapsulated inside. So if you look at the two vaccines, that is the Pfizer vaccine and the Moderna vaccine, one of them, the messenger RNA codes for the spike protein, and for the mRNA here codes for the S protein. So this acts as a delivery vehicle, and this is a very modern or recent development which came out very fast during the COVID period in 2022, 2020 to 2022, when it was developed very fast. So I want to just tell you one thing, which that in India, Professor A.N. Mehta, many of you will know, he's no more there. You know, he started doing this kind of work in the year 2000, very early in nanotechnology. He was working on things like trying to put Taxol is also a drug for, cancer, anti-cancer drug, and trying to make non-viral vectors. You know, viral vectors can be toxic. So he was trying to develop non-viral vectors in those days in 2000. So just on recollecting him, nearly 24, 25 years, I personally know him. He was my teacher in masters. And I remember it was one of his lectures that I was in 99 that I also decided to enter the field of nanotechnology. So he was working, if you go back and look, in the year 2004-5, he has several papers on looking at different aspects of stabilizing Taxol and many other drugs in different encapsulated medium. So like calcium phosphate is an inorganic material, and he showed that it is a non-viral vector for Taxol. But he has worked on very micelles. You know, micelles are like spherical objects which are made using surfactant molecules. In liposome, you have 2 layers of surfactants. In micelles, you have single layer. And in that also, you can encapsulate Taxol. And he showed that. And he showed that if you can design micelles, which will open up only when it reaches the cancer cells, etc. And so very early, he was doing this kind of work. Today, there are much more smarter delivery systems. This is an example of what is called a zipper peptide. And this zipper peptide in a liposome, where you have encapsulated drugs, and you have also introduced a zipper peptide, which is a leucine-based zipper peptide, just to change the temperature by few degrees centigrade from 37 degrees to 43 degrees, and the zipper peptide opens up. And when it opens up, you can release the drug which was inside out of the zipper peptide. So this is a thermoresponsive drug. Well, this marketed drug that using the Celsion company, it releases doxorubicin, which is also a heat-sensitive drug. So you can see that at 42 degrees, the heat-sensitive liposome delivers the drug very sharply. So if you move from at 35 degrees, the heat-sensitive drug barely is above the free doxil when the body temperature is 37. But if you take it up to 40, 41 degrees, the release is much sharper of the doxil, which is in the free native state. The amount available is much less. So this is again a thermoresponsive drug which was developed by the Celsion Corporation. So as I was talking about gold nanoparticle for use in cancer by using laser light, because by laser light you can heat gold. And this is gold-coated silica nanoparticles, which was FDA approved, I think one of the first FDA approved drugs in 2002, I think. And this has silica, which is coated by gold, and you can use it under laser light, a particular laser light. Depending on the size of the gold nanoparticle, you will be able to activate it under different light. So 5 nanometer gold, you see you can use near IR radiation, which is around 1050 nanometers. As you know, you don't want to use very high energy radiation. So you want to use near IR radiation, which is around 1050, 1100 degrees is good for biological systems. And so if you have 5 nanometer particles here, which is coated by, which is silica coated with this kind of gold nanoparticles, they can be excited by this 1100, 1050 radiation. There are other particles which are can be used using magnetic field. You can study them. This is one MagForce company which uses to study glioblastoma. So I won't go into detail, but just to explain that you can use thermoresponsive particles. You can use optically responsive particles like gold, and you can use magnetically responsive particles to study and diffuse cancer cells in different parts of the body. This is an example which is very popular from literature, that you have, suppose, a cancer in this organ. There's a solid tumor, and if you have a nanoparticle which is going through the bloodstream, but if you attach a magnetic nanoparticle and apply a magnetic field, you can bring the magnetic field near the solid tumor, and then you can increase the magnetic field. Once you increase the magnetic field, the magnetic particles will increase in temperature, and the temperature, local temperature, will kill the cancer cells. Okay, so this is taken from literature. There are other aspects of cancer cells and normal cells which people use, like the distance between the cancer cells and the ability of nanoparticles to come in between the cancer cells and move easily than healthy cells. These aspects are used when one designs nanoparticles targeting cancer cells. Then coming to MRI contrast agents and other agents which are used when one is targeting cancer cells. So you can develop particles which will give you a better contrast, either MRI contrast or CT contrast. So different kind of nanoparticles can be developed, which will give you a better contrast, either when you are using a magnetic system or you are using an X-ray system to image the cancer cells. This is, of course, showing magnetic hyperthermia. So you have cancer cells, and then you use the magnetic field and with the drug doxorubicin or without the drug, and see how it is changing. So these are different types of things that one can do with different nanoparticles. So you can image, I'll come to imaging later. You can also do something using microfluidics and nanofluidics, where you can use channels to deliver drugs. So these nano channels, if this channel is in the width of nanometers, then we call it nanofluidics. If these channels are in micron size, then we call it microfluidics. For making drug delivery through these channels, you need clean rooms, etc. And you need a certain kind of very specialized pumps and microscopes. So these pumps make these droplets move through these channels. These channels are designed, as you see, there are corrugated systems, etc. This is, you are now biologists or doctors working with chemical engineers and mechanical engineers and nanolithography people. They combine together to make this technology where you can make two blood cells interact, or two any kind of droplets interact which has a drug, and you can study. So you can see this kind of a shape of this channel is constricted here to make these two droplets. You can study single droplet movement, two droplet movement based on the design of such biochips. So these are called, like we have electronic chips in electronic circuits where electrons move. Here droplets move, or biological systems, you can have blood move or urine move, and you can target certain kind of molecules inside them by doing different kind of shining light on these, etc. Many things you can do with these. So one of the drug delivery systems shown here is that you can design channels, and these channels are of the diameters of microns, some 100 nanometers to few microns, depending on your ability to synthesize microchannels. And you can have reservoirs of drugs, and these can be programmed. And behind this watch are microneedles. But if when you are wearing it, the program will automatically release drugs in the body. This was shown for chemotherapy using adjustable electrophoretic pumping. So as you see, this technology needs the combination of a mechanical engineer with a nanotechnologist, with a biologist and a doctor to study this kind of technology. But this kind of technology is already known for 10, 12 years. And using this kind of microfluidics, we can study several other kind of devices. This is a silicon nanowire based chip where one can study some antibody antigen interaction, and that will change the current passing through the nanowire. Using that, we can calculate the concentration of some molecule which you are trying to detect, for which you have a particular antibody on the surface of the silicon nanowire. So this is one example I'm showing. So it's like a biosensor. So this biosensor is based for the COVID vaccine. And this was developed using a FET sensor. So FET is a field effect transistor. It is very popular in electronics, one of the simplest things. So you have two electrodes, and in between you have a droplet which contains this virus. But this virus has to sit on top of a molecule which you have already put on top of this surface on here. And once the virus sits on top, it will bring about a change in the current which I am measuring. So this change in the current will be, I can calculate the concentration of the COVID virus on top. Like that, you can have different antibody antigen interactions for different viruses, and you can have biosensors for that. This is a FET based sensor. You can have a colorimetric sensor. This is also for COVID vaccine, but the color changes because of aggregation of these gold nanoparticles when the virus is present. So I won't go into detail because I'm not discussing colorimetric detection too much in this. So this is another lateral flow assay one can do. You can see that there are 3 lines. In these 3 lines, I have got 3 different antibodies. And if I have a multiple combination of antibodies flowing through this, one of them binds here, right? One of them binds here, one of them binds here. Then I'll get 3 different lines, and they are specific to certain molecules. Each antibody antigen interaction is separate. And so from these lines, these are basically IgG, IgM markers, and one of them is a COVID marker. So this, using this kind of a lateral flow from a mixture of molecules, you can see which one is present through this, what is called a lateral flow immunoassay. So this I already explained. Using a FET-based sensor, you can have a change in the conductance or resistance, which will tell you the kind of molecule sitting on the surface. And this in INST, we had developed many such sensors based on myoglobin, troponin, etc., which are supposed to be markers for cardiac arrest, etc. This kind of biomems, etc., this is a device developed in South Korea, where these disposable diagnostic biochips were made, and this can be inserted into this handheld device, which will automatically read the blood glucose, the pH, the creatinine level, etc. And it's also Wi-Fi enabled, so it can be, this data can be transferred to a doctor, etc. So now coming, because the last, I think 15 minutes I have, I started at 2:40, but I'm already a little late. I'll go fast. Some examples of our work. So we made a silica nanoparticle, which was designed effectively. So you see the silica has spikes on its surface. Now, how do you do that? That's a chemistry. So the spiky silica is important because that will adhere to the cells very well. So this spiky silica, we put some, we want zinc nanoparticle. As I said, zinc ions, when released in the cells, can break the cancer cell membranes because of reactive oxygen species. So zinc-based molecules we added onto this. It is a zinc imidazolate 90. It's a one kind of metal organic framework containing zinc on a spiky silica surface. Okay, so that's this spiky silica nanoparticle with the zinc imidazole framework 90 with doxorubicin is the molecule which we use to study some cancer cells. Okay, this is the work of Manoj Kumar. He did his PhD jointly with me and Michael Yu in University of Queensland. So what you can see is that once you have the silica nanoparticle ZIF90 doxorubicin, you can, the loading capacity you can see. You can see that we can have the particles circulate in the system and release at acidic pH. As you see, the environment near the cancer cells is more acidic. So the particles sustain the normal bloodstream and can be available for much more time till they go to acidic environments. So that release is at 4.5 pH. And then you can study the cell viability. So you can see the silica nanoparticle ZIF90 DOX gives the least cell viability. Also, the IC50 studies show that the silica nanoparticle ZIF90 DOX needs much less IC50 compared to pure doxorubicin, which is a drug for anti-cancers, or ZIF90. So you need the silica nanoparticle and ZIF90 along with the DOX to give you the right or much more efficient behavior. Right. So this nanocomposite, what does it do? It helps cellular internalization. It allows high loading of doxorubicin through imine bond formation. Okay. So there's an amino group in this doxorubicin. This drug has an amino group, and there is an imine bond formation. That's the chemistry actually which we use. And important thing is that it releases drug at 4.5 pH, not at 7.4, which is the physiological pH. And we can show using characterization, using microscopy, etc., that how all this nanoparticle allows selective and enhanced release of doxorubicin, especially in acidic environment. So this is one paper which we published. Then another work I quickly will go through. We use lysozyme, and along with lysozyme, we use again a metal organic framework silica nanocomposite against gram-negative bacteria, so H. pylori. So this is a disease, it's a gastric disease, causes ulcer and all that, and it's very popular. Nearly 50% people in our area have this kind of disease. So why use lysozyme? Lysozyme is available easily, and lysozyme has a property of breaking the cell wall, and it breaks the glycosidic bond, actually 1,4 glycosidic bond. And so we use lysozyme and the ZIF8 silica nanoparticle together, which also releases reactive oxygen species. So if you can see that when you have the both the silica nanoparticle with the MOF, here the MOF is ZIF8. In the previous example, the MOF was ZIF90. There are two different metal organic structures. This is like, you know, material chemistry. And so the reason of choosing this MOF, there is a reason to choose this particular MOF along with silica nanoparticle. And we make rough silica nanoparticle to adhere to the H. pylori cells along with the lysozyme. And you can see that the reactive oxygen species generation is much more in this particular case. So these are microscopes to show you when you have the ZIF8, the particles are adhering to the cell walls, etc. And the cell viability is much less when you are using this HSNP ZIF8 lysozyme. These are on CACO2 cells, and we studied the concentration dependent of the cell viability. So the Heliobacter pylori, which causes peptic ulcer, etc. Normally people treat it with antibiotics, amoxicillin and clarithromycin, but it has been found that now there is antibiotic resistance. So can we have something which is not using antibiotics? So this is one example that we studied. Can we use a lysozyme and along with a silica-based nanoparticle to work against this H. pylori? And we got some success in that. Okay, so our formulation releases zinc ions in close proximity of bacteria and strongly inhibit bacterial infection. The biocompatibility of our nanoparticles on human cancer cell lines shows minimal cytotoxicity even at high dosages. If I have 5 minutes, I will talk about luminescent nanoparticles, which is another area where it's more for imaging. So we make nanoparticles of this kind. If you can see, this is using rare earth ions. You know, rare earth ions from chemistry we know have FF transitions which give out light. So this emission, once this particle is in the cell, it can emit light, which we can see for imaging. So the particle that you have to make should not be toxic. So we chose a sodium bismuth fluoride based particle, which is very new. And this paper we have published in PCCP in 2023. The biology work was done with Professor Perumal in IIT Delhi in the Biosciences department. So what I will just go, this is those who want to understand the optical properties or the physical chemistry part of it. We are using these transitions, FF transitions, and we are using a light which is in the near 1000 nanometers, 980 nanometers. As I said, we want in biology always to use higher wavelength light. So 980 nanometer, 1100 nanometer is useful, but the light that we want to see should be in the visible. So how to do that? We use what is called up-converted light nanoparticles. So the design of our nanoparticle allows, although we are using 980 nanometers, it will allow us to see in the green or in the red, which is at 500 nanometers, 600 nanometers, etc. So this design of the nanoparticles is the knowledge of physical chemistry, where you have to understand this energy levels, how the electrons will move from higher energy to lower energy and emit radiation in the visible region. So up-converted nanoparticle is a research which is very important in imaging. So this is in biology, the cell lines to show the viability. That means our nanoparticles are not doing any harm to the cells. This is what we want to show. And then we study the blue light is due to a drug, and the green is coming from our nanoparticle. So what it is showing is it is entering the cells, it's in the cytoplasm, and the drug which is in the nucleus is the other blue drug. So it shows that our nanoparticles are entering the cells. So if we want, we can attach a drug and it will enter the cell. So the imaging part is what we have done using this nanoparticle. And it also shows that the bacterial growth is not affected by our nanoparticles, etc. This is to show that this particular nanoparticle is useful for negative, the gram-negative bacteria, where from you can see the emission much better. But for gram-positive bacteria, it is not showing much emission. So it is useful for gram negative bacteria where we can see the emission. The other thing is similar thing we have done where we can show both. We have used it to image. Also, we have used it to kill the cells. So the blue here you can see in the presence of light, if you see red, that is the dead cells, and the green ones are the live cells. So when we are using our particles, these nanoparticles, as we said, these are different nanoparticle. This has got ytterbium ions and erbium ions, and the create this material, nanoparticles going into the cells, and then you are using some dyes, and what it shows that in the presence of light, if you are killing the cells because of these nanoparticles, and in the absence of light, there are more live cells. So we used a glioblastoma cell line, C6 lines, and also a normal mouse fibroblast line. 2 cell lines we used, and we can show that the cell viability really went down in this case. I don't have much time, but I can just show the last test. This is more for X-ray tomography. These are particles we are developing, which will be, when injected, gives you a persistent luminescence. That means you use X-rays for a very limited time, but the light which will come out from the cell, from the body, is much more delayed. So you don't want to use X-rays for a long time. You use only short pulses, right? But the decay, the luminescence should be there for a longer time. So this kind of particles we have made is just published in Journal of Materials Chemistry. See, this is with joint research with Professor Paras Prasad at University of Buffalo, where we are designing these nanoparticles, which will give you a persistent luminescence when embedded in a polymer. We have shown this, and we are going to take it to clinical trials. So I will stop here. I've given you some examples from our work of nanoparticles which can embed drugs and deliver, and nanoparticles which can be used for imaging. So 2 purposes. So with that, I come to a conclusion. Sorry, I took nearly 1 hour, I think. Thank you very much.
So it's pleasure and privilege for me to thank Professor Ganguly for very informative talk on a different aspect. This was a good interface. I hope it is very interesting for all of us as well. And there are some questions. So one is that this Dr. Manpreet Kaur says, hello everyone, but she is a food technologist. So she said, can we cover role of nanotechnology in food science? I think later on sometime again, we will have this kind of aspects in our global series. Second is from Indonesia, Dr. Rudy Nugroho is there. And he said, thanks for permitting to joining me this marvelous seminar. My name is so-and-so, and I am an animal physiologist, but interested in nanotechnology. Glad to know and be here. So, Professor Ganguly, these are good comments for you. And another says, this is Rita, says, brilliant lecture, such a vast knowledge and amazing flow. Thank you, Professor Ganguly, for enlightening us. And other are many thanks. I think each one is writing thanks to you. Mostly I'm seeing there are no question, but nice.
I must tell you, I am also learning because there is so much biology to learn. So I'm learning from my students.
No, for this society.
Yeah.
So for the society also, this aspect is rarely we dealt into. And I'm now seeing a number of, messages saying excellent and very informative, different aspects, and they all are thanking you. So if there is any other question for others as well, this was just chat box I was reading. If there are any questions from the participants.
साहब मैं एक बोल सकता है क्या पूछ सकता है क्या
Dr. Madhyastha.
Professor Ganguly, yes sir. I am Harish.
So
Harish.
Professor Madhyastha is from Japan, and he would like to ask a question to you, Mr.
Sure.
Ganguly. Yes.
Sure.
Nice presentation, Professor Ganguly.
Hmm.
In regarding the cancer diagnostic and diagnostic aspect, nowadays people are using, am I audible, sir?
Yes, yes.
Nowadays people are using diagnostic with the foliate reagents, folic acid receptor based this one. What is your say on this, sir?
So as I said, I am not really an expert
Okay,
cancer
sir.
biologist. So, but I have seen people using folate, etc. And I'm sure some biologists can answer why they are using. So, but I
No.
will not be able to answer.
A cancer cell has the folic acid receptor.
So they must be targeting that receptor. They must be
Yes,
using
but
molecules on the liposome which will target that folate receptor.
right,
That
sir.
is
But
one way to do it.
what happened actually is during the autolysis and lysosomal capture, encapsulation and release mechanism, this separation of the drug from the metallic conjugated or some other conjugated system not takes place inside the cytosols level.
Yes, yes.
This
True.
is my experience. This is my experience.
Yeah. So whatever little I know, there is this endocytosis and this endosomal pathway is there. And once the endosome forms, then there are 4 or 5 mechanisms. There is something called proton sponge mechanism and
Correct.
some other mechanisms. I'm not an expert, but whatever I hear when I discuss with biologists, so these are different mechanisms. And I believe some of these are working. In our particular case, one paper which we have written, there this proton sponge mechanism is working. We could prove that.
Yes. Thank you.
Yes, so thank you.
There are a lot of questions, Sunilji, in the chat box. Can you take up?
Yeah, I can now see question coming up. So
Yeah.
I was about to say that. So one is that can we kindly have something covering synthesis and application of green nanoparticles from microalgae or otherwise? I think Professor Ganguly, you need to come again in the main BRSI.
Yeah, there are a lot
cover
of
these
work
aspects.
actually. A lot of people are working for what is that, green nanoparticle synthesis.
And other is, can we detect nano contamination in environment?
Oh, yes. There's a lot of work going on that. Nano contamination in the environment, in water. There's a lot of work on that. Lot of work.
And there is another question on quantum dots in imaging and toxicity and their toxicity.
So that I can tell you. That is why, see, we are imaging using certain particles. I can actually image much better using quantum dots. But quantum dots are kind of toxic. That is why people don't want to use. So they want to use other kind of particles. So this is the particle like we have discussed here, sodium bismuth fluoride and doped with some rare earth is not toxic. But their intensity of image, that the light coming out, would be much less than if I use a quantum dot like cadmium selenide, cadmium sulfide. They are much powerful that way. But the toxicity prevents us from using such quantum dots.
Okay, so thank you. And there are more questions on biologically synthesized green nanoparticle. I think, as you said, there are a number of work available using algae also. One can refer, but maybe you can talk to call to and interact with Dr. Ganguly. If you go to ISAR Behrampur website, you'll find his contacts there individually.
Although I don't directly work, but I can give you details.
Yeah. And then there is last Shrey Dubey, his future of green nanoparticle. Again, same thing, Mr. Dubey. I would like to say that you can interact with him more on this. Any further question? If not there, sir, you would like to say?
Before I said that, can you stop sharing the screen, Professor Ganguly?
Yeah. Let me stop here.
I would like to invite Professor Christian Laroche, administrator of IBA, to say a few words. Christian, are you there? If not, Mohammed Tahir, Mohammed, are you there? He's a coordinator from IBA side for this global lecture series. Mohammed? I think they have left.
Because of different time zone, I think, for them.
Yeah, they were there, but yeah.
I
So,
saw it.
Professor Ganguly, that if anybody has any other question, Alok, if you have anything to say, or Smita, you have anything to say? Otherwise, I'll just move to the.
I think it was a fantastic talk, sir. Congratulations, Dr. Ganguly.
Thank you very
Very
much.
informative.
Very interdisciplinary. But you know,
Yes.
I'm enjoying learning biology. There's so much to learn.
So, Professor Ganguly, one of the Professor Krishna Mishra, you have anything to say?
Yeah, just I have no question. I mean, it was wonderful lecture, and I just want to know a general thing because nanoparticles, they say that they have cytotoxicity. I mean, as compared to normal drug delivery, the nano size of drugs, do they have more toxicity?
No, actually,
I mean,
see, that is the research people
comparative-
have done in the last 10, 15 years. That is what size of nanoparticle to be used, what kind of material you should use, metal nanoparticle, if metal, then which one? Like gold can be used at a certain size. Not all size can be used. Then preferably people are going into other, say, silicon nanoparticles. So they are doing a lot of research, and it is not that every nanoparticle is toxic. You have to use the right kind and the size, because size and shape matter, because otherwise they can go into different organs of our body. But you need nanoparticles which will be cleared after the cargo is delivered, or which will be degraded and excreted. So that is the main research, and that is what people have done. And there are now several nanoparticle-based drugs in the market, FDA approved.
Okay, I mean, they say that due to their small size, they can enter any normal cell, so that's how they
Yes.
create more.
Yeah, so due to their small size means, again, you have to be very specific about the size range. So that's what I gave a picture. Certain size, certain shape will get cleared very easily. Certain shape will go and, target the liver cells and will be difficult to move. So that kind of now, a lot of data is there. Which type of nanoparticles has to be used. So what I showed you, that was all the design is because of all the chemistry one has to do is to see that the nanoparticles are not toxic. They are, or if they are metal, then they should be easily excreted out of the system. So these are today, lot of information, etc., is known in poly.
Okay, thank you, Dr. Ganguly. I just one question. I mean, they say that TEM is the most sensitive this thing for measurement of homogeneity of any nanoparticle. I mean, as compared to DLS and SEM.
TEM. No, TEM is depending on which kind of TEM, you can get a more accurate size, small, especially at small sizes. If you are looking something which is 1 nanometer, 2 nanometer, then telling precisely that whether it is 2 nanometer or 1 nanometer or 1.5 nanometer, you have to use a high resolution transmission electron microscope. Whereas SEM can see much larger areas, but there may be, you can see something like 10 nanometer, but the error in 10 nanometer may be 2 nanometer or 1 nanometer. So you may not be able to say 10 and 10.2 nanometer, but in a very advanced transmission electron microscope, you may be able to tell 1 nanometer and 1.2 nanometer difference.
Okay.
More precise.
Okay.
You are, I think these nanoparticles may be used for pro diagnosis of cancer. That will be.
Yeah, nanoparticles are being used, as I said, a lot for imaging. And if you can do imaging as well as drug delivery with the same probe, that's what you don't want, several probes in the body. So you want one which can image as well as do the drug delivery. So those are the kind of advanced techniques people are using. And all this research now, nanomedicine research, everywhere doctor is involved. You cannot be just a materials person or just a biologist. You have to have a doctor with you in your research team. So
Thank you. Thank you.
So, Professor Ganguly, that one of the questions which I normally ask is not on the topic of your presentation today. Is BRSI as well as IB have a lot of young budding researchers and people slowly have understood much more relevance of interdisciplinary research. This is what we have very well exemplified from your own work that coming from material chemistry and its application in the medicine and biology. This is something fantastic. So what message you would like to give to the members of our societies, especially to the young researchers or who are thinking that how to move forward, to do outstanding quality of research and so on?
Yeah, so one thing is, see, all the major challenges which are lying will all happen at interfaces. Either interface with biology, with physics, biology with mechanical engineering, biology with chemistry is very old. We are studying biochemistry for a long time. But biology with electrical engineering, biology with mechanical engineering. Earlier, people used to study electrophysiology, you pass current and see things changing. Now it can be electromechanical biological system. That means mechanical stress with electrical charge flowing, what is happening to the cell. So these are things which already 10 years, 15 years old, there are a lot of scientists in the world. And if you look at Google, all the top scientists in the world who are getting billion-dollar projects, they are all working on topics which are at the interface of subjects. And very, very interesting things are happening at the interface. For example, the cell, a biological cell, whether I can hold it with a laser beam. These people studied long time back. It is called optical tweezers. And I can hold even one cell with two lasers and squeeze the cell. And what is the flexibility of the cell? So these are things which people studied even in 2005. There are papers in Biomaterial. There's a journal by Subra Suresh, who was at MIT. And so there are a lot of things such which today's young people see. In 20 years back, maybe one person who has that kind of idea was working, unusual idea, Today, you will have many people in interdisciplinary research. So think of new ideas, new interfaces of subjects where things will be difficult for you because it will be challenging. How will you, say, imagine 20 years back, how will a mechanical engineer think I will study a cell and try to hold it with a laser? But they did that, and they studied not only one cell, a system of cells. Like they could tell exactly in a colony of cells, how many cells have what kind of flexibility of their cell membrane. And these are complex problems, but one throws up challenge and then they can study.
Thank you so much. Thank you so much, Mr. Ganguly. Over to you, Sunilji.
Yeah, so it's time for me to thank Professor Ganguly on my personal behalf and on behalf of the BRSI. Indeed, we appreciate your sparing valuable time for us, and also that you gave such an informative talk and saying that interface is what is coming more and more. I think that message will be sent across to all participants, especially the young researchers. And also for us, it was lots of excitement to listen the interface between nano and bio. So thank you again. I think, and so one more announcement before we close is that the next 16th BRSI Global Lecture Series lecture will be held on 18th September at 2:30 p.m. IST by Professor Mohammad Tehrez from Sweden. So this is, we will circulate it later, but just to share that. And I also thank Professor Pandey and other distinguished participants and students and member of the society for giving time and attending this lecture, making it a success. Thank you, everyone. Thank you very much.
Thank you.
Yeah.
Thanks, everyone.
Everybody.
Thanks. Thanks.
Thanks.